Okay, hello everyone and welcome to the new video to the chapter on the nucleic acids, and today I'm gonna talk to you about the general structure of nucleic acids, and I'm gonna specify those information on the structure of DNA molecules. So let's start with the phenomenon of nucleoproteins. So if you remember, or if you have watched some of the previous videos, I have described complex proteins or heteroproteins containing both protein part of the molecule and different molecules which are defined by the term prosthetic group. So when we are talking about nucleoproteins, we can say that they are complex proteins or heteroproteins where prosthetic group is actually nucleic acid. So let's start our story today with the basic and general structure of nucleoproteins. Nucleoproteins are named upon the fact that they have been found in a nucleus first, and the Latin name for nucleus is nucleus. And apart from the fact that they have been primarily found in nucleus, they can also be found in cytosol and in certain organelles. So in ribosomes you can find some RNAs, and in mitochondria you can find some additional DNA molecules. So their name actually originates from this site where, or from the point where they are found when they were found first and they were discovered in nucleus first. And really, this is just one of the informative facts that nucleoproteins were first isolated from the nucleus of salmon semen cells. And nucleoproteins were isolated first by Friedrich Mischer in 1869. The major role of nucleoproteins is definitely something that we all know very well, and that is the transfer of hereditary information. And also what we know and what we have already mentioned in the chapter on proteins, nucleoproteins, DNA and RNAs are actually involved in protein synthesis. So let's go through the general structure of one nucleoprotein. So nucleoprotein consists of the protein part of the molecule and the prosthetic group, which is nucleic acid. So the protein part of the molecule actually consists of histones and protamines. I think that you remember and that you have heard about these names. I have mentioned them in the chapter on proteins, stating that they belong to the group of simple or homoproteins containing only amino acids in the structure. And I emphasized at that time that I'm gonna discuss a little bit more about their biological function when the time comes. And this is one of the spots where is the time to mention them, and that is that histones and protamines are in the structure of nucleoproteins as the protein part of the molecule. Those two types of proteins exhibit rather basic or alkaline character due to the presence of a significant amount of arginine and lysine in the structure. But there are a huge number of other proteins as well, which are found in the structure of nucleoproteins, which are actually tend to form the chromatin moiety. Non-histonic proteins are predominantly those belonging to the group of gene regulatory proteins, and they are involved in the regulation of the process of gene expression. On the other side, the prosthetic group actually exhibits a pronounced acidic character, And prosthetic group is, as we have already mentioned, nucleic acid. So if we have a protein which exhibits alkaline properties, and if we have nucleic acid which exhibit acidic properties, one of the possible conclusions is that those two components are gonna stick together by the formation of salt type bonding. If you take a look at the figure here on the slide, you see the review of these facts. So nucleoproteins are complex proteins containing the protein part of the molecule and nucleic acid as the prosthetic group. Protein part of the molecule predominantly consists of protamines and histones, and nucleic acid prosthetic group can be either DNA or RNA molecule. So let's explain the structure of one nucleoprotein. So nucleoproteins have very specific characteristic tertiary and quaternary structure. You know the definitions of those two types of and levels of protein structure organization. So in the structure of one nucleoproteins, 5 types of histones are involved, and those types are marked as H1, H2A, H2B, H3, and H4. We have mentioned that they exhibit basic character and they are positively charged molecules, and they tend to form ionic bonds with negatively charged nucleic acids carrying acidic properties. So what is the quaternary structure of those? It's very specific, it's very characteristic. So one of the moieties of the complex nucleoproteins is nucleosome. Nucleosome actually represents and is formed upon the association of nucleic acid with histones in a very specific and strictly defined way. In eukaryotic cells, and that is what we are interested in, actually one nucleosome consists of 2 monomers, and each monomer contains I'm sorry, 8 molecules of histones, types H2A, H2B, H3, and H4. So 8 of those molecules, they tend to form octamer. And that octamer is actually the core or the nucleus of the future nucleosome. You can see it here in yellow-green shaded globular structures. So we have 8 histones forming very specific octamer, and 2 of those monomers are actually forming nucleosome. What else do we have? If you see from the figure, we have DNA strand, which is actually wound nearly twice around this nucleosome core. And between those 2 monomers, we have a short sequence of DNA nucleotides, 50 to 80 base pairs, connected or stabilized by the action of one other histone, which is H1 histone. So this short sequence of DNA is marked as linker DNA or spacer DNA. So 2 monomers plus linker DNA and H1 histone, they tend to form nucleosome. And each nucleosome consists of 2 monomers, and each monomer of 8 histones forming octamer and 2 folds of DNA strand. Let's move on. But each DNA strand actually contains is a polymer of basic building bricks which are marked as nucleotides. So building bricks of DNA and RNA of nucleic acids are marked as nucleotides, and when they polymerize, when they're attached to each other, they tend to form polynucleotides. That is analogous to the story on proteins. So we had amino acids as building bricks, forming polypeptides as polymers, and here we have nucleotides as building bricks, forming polynucleotides as polymers. So nucleotides are rather complex structures, and they contain actually three groups. The first group is heterocyclic nitrogen base, which can be a derivative of a purine or pyrimidine ring. We will see it in the upcoming figures. Then it contains sugar component, which is always a pentose and aldose, ribose, deoxyribose. And the third component of each nucleotide is phosphoric acid or a phosphate group attached, as you can see from the slide, to the sugar moiety of one nucleotide. So let's go through the structure of one nucleotide one by one. So let's start with nitrogen bases, then we will move to pentoses, and we will finish with the explanation of the bonding of the phosphate group to the sugar moiety. So heterocyclic nitrogen bases are, as I have said, derivatives of purine or pyrimidine ring, and the structure of those two heterocyclic rings is presented here in the figure. And those are planar molecule, because if you remember your chemistry course, those conjugate double bonds tend to form planar structures. So these condensed rings are planar. So purine ring is planar, pyrimidine ring is planar. And different groups, atomic groups, can be attached to those two rings. So we can attach amino group, Na2 group. We can attach keto group, double bond O group, if you remember. We can attach methyl group, CH3 groups, and so on to these heterocycles. Each of the nitrogen bases actually has its UPAC official chemical name, but more often trivial or common names are used in everyday practice. They are also marked with symbols, abbreviations, which are actually the first letter of the trivial common name, and actually that is the big capital letter. So purine nitrogen bases, which can be found in the structure of nucleic acids, are adenine and guanine, and here you can see the structure of those two. The first one is 6-amino-purine, and the other one is 2-amino-6-oxypurine. So those are their chemical names, and the abbreviation for adenine is capital letter A, and the abbreviation for guanine is the capital letter G. But apart from those two nitrogen bases, which are found in the structure of DNA and RNA, we have some other purine bases which are intermediates of metabolism, and those are hypoxanthine, which is 6-oxypurine, then xanthine as an oxidized form of the previous one, 2,6-deoxypurine, and uric acid as the oxidized form of the previous one, which is 2,6-A3-oxypurine, and uric acid is actually the final degradative product of the metabolism of adenine and guanine, and that is the molecule which is going to be excreted from our organism. But anyway, let's move on to pyrimidine nitrogen bases. Three types of pyrimidine nitrogen bases can be found in nucleic acids. Cytosine, which is 2-oxy-4-aminopyrimidine, marked with capital letter C, and you can see the structure of those pyrimidine nitrogen bases. Then thymine, 2,4-deoxy-5-methylpyrimidine, so we see the situation where a methyl group can be attached to the heterocyclic ring, in this case pyrimidine ring. And we have the oxidized form of it, minus methyl group, so uracil, which is only for 2,4-deoxypyrimidine, or marked with a capital letter U. So what are the roles of nucleotides? I have made a list of a few of those. I couldn't make all of the list, but nucleotides, and this is something that comes to everyone's mind at the first place, so nucleotides are components of DNA and RNA. So they are involved in the growth of the cell, in the cell division, in the proteosynthesis, and all of those processes are going to be elucidated during this biochem course, but of course later on. Also, they are components of one specific group of molecules which are marked as coenzymes, and those molecules are actually often required for the proper action of enzymes, and they are involved in the catalysis of chemical or biochemical reactions in our body. And some of the examples of coenzymes which have the nucleotide structure are NAD, nicotinamide adenine dinucleotide, so the name says itself that it is nucleotide or dinucleotide. Then flavin adenine dinucleotide, again we have one dinucleotide containing some nitrogen bases. And coenzyme A, also containing some nitrogen bases in the structure. Also nucleotides and nitrogen bases, they are active intermediary compounds in metabolism, and I have provided you with one example, uridine diphosphate glucose, which is an activated form of glucose by which glucose can be involved in numerous biosynthetic pathways in our body, and those activated forms of different metabolites or different biomolecules are very important. We're going to talk about it in details, but when the time comes in the chapter on metabolism in the video and in the videos on metabolic pathways. Also, they are components of high energy compounds like ATP, CTP, GTP, and UTP. ATP is, I think, the most famous of all of them, because the energy released during catabolic degradative processes is actually captured in the form of ATP, and then ATP is used as energy donor for numerous biosynthetic processes. So anyway, nitrogen bases are also the ones which can enable activities of numerous hormones or growth factors or cytokines. They are involved in the phosphorylation of proteins, and for example, phosphorylation and dephosphorylation of enzymes is one of the very important pathways of activating and deactivating certain metabolic processes, and we will come to it in the upcoming videos and chapters. They can also be inhibitors of nucleotide synthesis. For example, due to mutations, you can have dimers of thymine, which will disable the formation and synthesis of nucleic acids, or some modified nitrogen bases like are used in cytostatic, so they are used in the therapy of malignant diseases because they exhibit some anti-proliferative activity. So there are numerous roles of nitrogen bases, both purine and pyrimidine, but in a way let's move on to see which molecules are attached to those nitrogen bases. So the second component is sugar component, and 2 pentoses are involved in the structure, that is D-ribose, or specifically beta-D ribofuranose is involved in the structure, beta OH group above the ring of the pentose, and D-deoxyribose is found, which is beta-D2'-deoxyribofuranose, the full name, and the structures are represented here in the figure. So this is beta-D ribofuranose, and this is 2'-OH group, which deoxyribose lacks. And that is the only difference, between those 2 pentoses. So D-ribose is found in the structure of ribonucleotides, and they are building bricks of ribonucleic acids, RNA, and D2-deoxyribose, it tends to form deoxyribonucleotide, and deoxyribonucleotides are found in deoxyribonucleic acid or DNA. When a nitrogen base attaches the sugar component, so then the nucleosides are formed. So nitrogen base plus sugar component, nucleoside is formed, and the bond which is formed by attaching the sugar component to the nitrogen base is actually formed upon the reaction of a nitrogen base and pentose. Nucleosides are formed, and the bond which is formed is marked as N-glycosidic bond. So by the definition, N-glycosidic bond is formed in the reaction of the semi-acetal or hemiacetal OH group from C1 atom of the sugar component, and you can see it here. So this is the hemiacetal or semi-acetal OH group at C1 of the sugar component of the pentose, and hydrogen atom at nitrogen number 9 or nitrogen number 1 of pyrimidines or of pyrimidine bases. So here we have purine nucleosides, and the name of purine nucleosides is formed by the addition of a specific suffix osine, O-S-I-N-E. So adenine becomes adenosine or deoxyadenosine if it's in DNA, or guanosine or deoxyguanosine. And here you see the structures of adenosine and guanosine, and this specific 1,9 N-glycosidic bond in purine nucleosides. In pyrimidine nucleosides, N-glycosidic bond is formed between the semi-acetal OH group from C1 of the pentose and hydrogen from N1 atom of the pyrimidine base. So it's a specific N1 glycosidic bond, and those nucleosides containing pyrimidine bases, their names are formed by adding specific suffix idine or I-D-I-N-E. So from cytosine we have cytidine or deoxycytidine. Then we have only deoxythymidine because it can be found only in DNA molecules, and uridine, only uridine with ribose in the structure because it's found exclusively in RNA molecules. This figure at the bottom of the slide represents the structures of those pyrimidine nucleosides. Let's move on. When the third and the last component is attached to the sugar moiety, then we have the formation of nucleotides. Then the nucleotides are formed. So phosphate group is attached by ester bond to the hydroxyl group at the position number 5' of pentose, as you can see here from the slide, thus forming a nucleotide which name is formed as a nucleoside 5' phosphate. Nucleotides can be formed to bind 1, 2, or 3 phosphate groups, thus forming mono, di, or triphosphates. But be very careful here. Notice that the first phosphate group is attached to the sugar component by the formation of phosphoester bond. The second and the third phosphate group are attached to the previous phosphate group by the formation of phosphoanhydride bond. And those 1 or 2 phosphoanhydride bonds are high energy bonds. Thus, ATP molecule, adenosine triphosphate molecule from this figure is considered to be a high energy compound, but adenosine monophosphate containing only phosphoester bond and one phosphate group is marked as low energy compound. So let's sort the names. So from adenine we have adenosine 5' monophosphate, diphosphate, triphosphate with abbreviations AMP, ADP, ATP. AMP is also known by the name adenylic acid or adenylate as the form of salt. Guanine, the situation is analogous, as you can see. Guanosine 5' mono D3-phosphate, GMP, GDP, GTP, or guanidylic acid or guanidylate. Then pyrimidine nucleotides are formed by the attachment of phosphate groups, 1 to 3 phosphate groups to the nucleosides. So from cytosine we have cytidine 5' mono D3-phosphate, CMP, CDP, CTP, or cytidylic acid instead of CMP. Then thymine is always deoxythymidine 5' mono D3-phosphate, or DTMP, DTDP, DTTP, or DTMP is known as deoxythymidylic acid. And finally uracil tend to form uridine 5' mono D3-phosphates, UMP, UDP, UTP, and UMP is also known by the term uridylic acid. What can we say about DNA molecule itself? It is a very well-known fact that there are two types of nucleic acids, deoxyribonucleic acid, DNA, and ribonucleic acid, RNA. So DNA consists of bases A, G, C, and T, so it contains thymine, and pentose is deoxyribose. RNA molecule contains bases AGC, but this time uracil instead of thymine, and pentose component is ribose instead of deoxyribose. So binding of the two nucleotides is achieved by the formation of covalent 3', 5' phosphodiester bond, which is formed, as you can see from the slide, upon the reaction of the OH group here at the position C5 of the pentose, and OH group from the C3' OH group from another molecule of nucleotide. So two phosphoester bonds are formed, and we say they are 3', 5' phosphodiester bonds. What else do we know? That there is an OH group from the phosphoric group, from the phosphate group, at the 5' end of an atom. So at the 5' end of a DNA strand or chain, we have a phosphate group, an OH group from the phosphate group. So it's marked as 5' end or phosphoryl end. But on the other side of the molecule, we have free OH group at C3 carbon atom of the pentose. So it's marked as 3' hydroxyl end. So this is reviewed here. What else can we say about one strand of DNA? So if you take a look here at the figure, you can notice two domains of the strand. So the first one is the core of the DNA strand, having phosphate and sugar components, in this case deoxyribose components, phosphate deoxyribose, and so on. And this linear part of the molecule actually represents the so-called chain backbone, or the core of the chain. So those are intermittently attached phosphate groups and pentoses. And nitrogen bases, if you notice from here, from both figures, are actually stacked inside, and they are oriented perpendicularly to the axis of the chain. So, and this is the last information for you. So 5' end, we have already mentioned free phosphate group, and 3' end, we have free hydroxyl group. So you can see it from the bottom figure as well. So at the 3' end, we have free OH group from the ribose or deoxyribose, and at the 5' end, we have a free phosphate group. So, the secondary structure of DNA is described by Watson and Crick in 1953, and for their discovery they have been awarded the Nobel Prize. So they have proposed the complex structure of the DNA molecule that is familiar to us nowadays. And they suggested that the DNA molecule is actually the double chain helix, so that it consists of 2 polynucleotide chains which are coiled right-handed around the common axis. Also, they have found that those 2 chains are anti-parallel. So the one chain goes, and this is here is the figure. So the one chain goes in the direction 3', 5', and these are the credits from the sites where I borrowed these. And then from 5' to 3' is the second strand. What else did they have described? So the chains are anti-parallel. So 5' end of one strand is paired with a 3' end of another strand, or opposite. Also, they noticed that those chains are not identical but rather complementary, which means that opposed to adenine in chain 1, we have thymine in chain 2, or opposed to guanine in chain 1, cytosine is found in chain 2. Also, they have proposed that between adenine and thymine, 2 hydrogen bonds are formed, and between guanine and cytosine, 3 hydrogen bonds are formed. So let's review this. So here are the base pairing rules. So due to the fact that those 2 strands are anti-parallel, for example 5', 3', 5', 3' in another direction, so the nitrogen bases obey the base pairing rule or the complementary rule. So this is thymine attached to adenine with 2 hydrogen bonds, and here we have cytosine and guanine stabilized by 3 hydrogen bonds. What else do we know? This specific structure of DNA molecule is actually a chemical base of the heritage of all cells. And we can also divide the structure of the DNA molecule into 2 domains. So purine and pyrimidine bases, they actually tend to carry the genetic information, but the sugar and the phosphate residues, they rather exhibit structural role. So the type and sequence of nucleotides in the DNA, it represents the primary structure, like it was the sequence of amino acids in a protein, and it's specific to each species. It is important to emphasize the fact that the variations and variability of DNA is rather huge, and every one of us has its specific DNA sequence or nucleotide sequence in our DNAs. Those parts of the DNA molecule, linear parts of the DNA molecule, are marked as genes. So, what else Watson and Crick proposed? That the overall structure actually resembles a twisted ladder, and the spatial relationship between 2 strands in the helix creates a major or wide group and a minor or narrow group, and this is groove, and this is what you can see here. So this is the major groove and this is the minor groove here. And of course there are some dimensions of specific regions in the DNA molecule. You don't have to memorize all of these numbers, so these are just for information. So diameters of helix is the same throughout the DNA molecule. And if you notice from the slide, so purine and pyrimidine bases are stacked inside, so and they represent actually the hydrophobic part of the molecule and of the helix, and it's perpendicular, as I have already mentioned, to the axis of symmetry. And phosphate group and pentoses, as you can see here, they are oriented to the towards the outside, to the environment, and they are actually in the contact with the aqueous surroundings due to the negative charge on phosphate group. So the coil diameter here, so the coil diameter is 2 nanometers, and if you can see from the figure, and the distance between two bases is 0.34. Here it is. The distance between bases is 0.34 nanometers. And also what you can see here from the figure is that approximately 10 bases tend to form one round of a helix, so the length is approximately 3.4 nanometers. And these are not all the facts about the DNA molecule, so these are what Watson and Crick proposed, but later on some other scientists, they tend to establish some new additional rules regarding DNA molecule, and one of the famous rule is marked as the Chargaff's rules by the scientist Erwin Chargaff, who posted those rules, and they say the following: ratio A over T is 1 and G over C is 1, which means that the number of adenine molecules is equal to the number of thymine molecules in DNA, and it is the same for guanine and cytosine. Their quantities are equal. The second rule is that the number of purine nucleotides is equal to the number of pyrimidine nucleosides due to the base pairing rule, which means that A plus G over T plus C equals 1. Some of these rules are not applicable to RNA molecules, so they are valid for DNA molecule only. And a couple of other terms. If you remember, we have mentioned that if a protein molecule is exposed to the activity of certain or the effect of certain factor, denaturation might occur, and also with the DNA molecule as well. So the secondary structure of DNA molecule may undergo denaturation process, which is actually described by the depletion or the loss of the secondary structure. Predominantly the double helix is unwound or splitted into two separate strands. That can be achieved by the addition, by increasing the pH of the solution towards the alkaline, highly alkaline medium, or by the increasing the temperature. And there is the fact that the more AT pairs in the molecule, the lower the temperature of denaturation. But oppositely to the denaturation of proteins, which was irreversible, and it terminates all the structural properties and the function of that specific protein. In this case, under the adequate conditions, renaturation is possible, meaning that the double strand and the double helix can be reconstituted upon the removal of those factors which actually caused the unwinding and denaturation. What is also interesting, that denaturated DNA molecule can be combined, or we say can be hybridized with some other molecules or parts of DNAs or RNA molecules, and that phenomenon and that procedure is marked as hybridization. So by the process of hybridization, you can get recombined molecules of DNA, and the phenomenon and process of hybridization is actually founded its wide application in molecular biology and genetic engineering, which is definitely the topic for absolutely another chapter. So more or less, that would be the basic information regarding a DNA molecule. And until our next chapter, our next video, in which I'm going to talk about RNA molecules, please stay safe, stay healthy, and bye-bye.